Di ba ibigay mo rin sa dito, hindi ba? Yung live mo. Para sa mga comments, di ba? Yes, okay mo.
How?
Which will be?
Good morning, everyone. For those who are already there, please say something. Happy New Year to you. How was your holidays? Can I see? Anong delay niya? For those who are already around, let's just wait for a bit until everybody is in. If you are there, if you are in, kindly type your name so that I can get your attendance. Hey, good morning, Christine, the earliest bird. Oh, hello, Maricar. Good morning and Happy New Year to you. Hello, Romy. Good morning. Hello, Almira. I like it. So let's just wait for others, okay, before I start the lecture so that we can maximize participation. Yeah. So how was your vacation? Nag-enjoy ba kayo? Maraming beach, maraming food. Hello to Kim Bernadette, Almira, Kimberly Joy, Shai May, Jillian, Via, Angela, Yen, Jamaica. Happy New Year, everybody. Hey, good morning, Karen. Can everybody hear me okay? Good morning, Cerilina. Good morning, Jaisel. Loud and clear. Oh, that's great. We'll just give everyone a little time for them to tune in before we start the lecture, okay? We'll start our lecture at exactly 9:40. While waiting for others, can we have a little fun? Can someone please share their number one New Year's resolution? Let's see what you have. Can anyone share with us? I'm sure that you do have your New Year's resolutions. So give us one, your number one in the list. Okay, we're waiting. Nobody would like to share? Maricar said, maraming food pero wala pong labasan ng bahay, ma'am. Oh, that's good. Staying at home is always the safer way. Asan na yung iba? 18. Wow, Maricar, magpapayat. That will be good. Si Christine, healthy living na ma'am. More veggies, less meat. I like that. I like that. Lalo kang gaganda niyan, Tin. Si Maricar din, lalong gaganda pag... Well, actually, okay lang naman kahit hindi payat. Pero for healthy purposes, go for it, Maricar. O yung iba, wanna share? Ano si Jenny? Wala na? Nobody else would like to share their New Year's resolution? Just bear with us. Please wait a bit. Other students are having problem tuning in. Hindi daw sila makapasok. Bakit kaya? Can someone please help your classmates? Daw sila makapasok. Bakit kaya? Okay. We're now 28. Good morning, Princess Jane. Good morning, Chevin. Good morning, Sophia. Marshall. Ano, New Year's resolution? Iwas love life? New Year's resolution ba yon? O sige, go for it. Via Angela, maagang gigising sa umaga. I like that. Then you do exercise, catch the morning sunlight. And Marshall, sa study. Oh, focus sa study. Sige. Shara, good morning. Micaela, good morning. Jillian, less puyat, ma'am, hopefully. That's good. That's healthy living as well. Tingnan natin kung less puyat talaga. We got 3 minutes. We're still waiting for others. Can you please help them? I hope while watching you also have your pen and paper so you can take down notes. Some of the things that we are going to discuss are actually in your post-tests. Hey, Happy New Year, Ernel. Happy New Year, Ray. Good morning. Good morning, Rufa. Jaja, tumaba na sana ngayong 2021. Ay, iba rin. Iba rin ang gusto, tumaba. Huwag lang pasobra, ano. In today's lecture, we will be joined by hopefully the Dean of the College of Fisheries and Aquatic Sciences for class observation. So, this is in preparation for the upcoming accreditation for BS Aquatic Biology and other programs. So it's 9:40. We got 33 people watching. I guess we can now start with our lecture. Okay. Others are still requesting. They need more time daw because the internet is a bit slow. But that's okay because they can always replay this video after we stop streaming. So let's start now with our discussion. So stay tuned until the end. Our topic today is actually DNA replication, transcription, and translation. So, we already know this, that DNA is the genetic material of all organisms on Earth. So, when we say genetic material of all organisms, that's for the organisms that we considered living. Don't think of the virus yet, because there are viruses which are RNA. But they are not really considered living organisms unless they are inside their hosts. So when DNA is transmitted from parents to offspring, it can determine some of the offspring's characteristics, such as their eye color or hair color for human, and flower color for plants, and antibiotic resistance, for example, among bacteria. So, what about you? Whose eye color did you get? Your mom or your dad? Or skin color, hair color, or the type of hair? Is it curly? Is it straight, wavy? Something like that. And that is determined by our genetic material, the genes that is embedded in our DNA. But how does the sequence of a DNA molecule actually affect human or other organisms' features? So the answer to that is genes specify functional products, specify functional trait that can be exhibited by the organism. For example, given in this, in our figure here, so we have the flower color gene, no? This is the gene that is governing the color of the flower. And then you have here the different sequences, the DNA sequence that would command protein that would help making the pigments of a particular color. So for example, here we got a purple color. So in other instances, you see white color, red color, and things like that. So this is just an example. Among microorganisms, this is also true. For example, a particular strain of Escherichia coli is antibiotic resistant. Okay, so there is a gene that governs antibiotic resistance, and that gene will command the making of the protein, making of the sequences that would protect itself from the antibiotic. So things like that. And then we have a DNA molecule being a strand with many genes. So for example, E. coli. When we think of E. coli, sometimes we say, oh, it's a single cell, it's a prokaryote, so it must have only a few genes. But E. coli actually can have as many as 3,000 genes. How much more human beings and other higher forms of organisms? So when we talk about this, we talk about the DNA, the genes, and making of proteins. These are actually the concept of the central dogma of life, and it is summarized in this figure. So from the DNA, which contains the gene, it takes two steps, the transcription and translation, to carry out a gene protein building instruction. So we will start from the DNA, and then what happens if the DNA is transcribed? So from being a DNA, it will be converted into RNA. So that's what is done in transcription. So you get a copy of the message from the DNA in form of messenger RNA. So it's an RNA. But in translation, when this one occurs in the cytoplasm of the cell, it just simply means that the message that is embedded in the messenger RNA will be translated into a chain of polypeptide making a protein. So eventually, in the central dogma of life, our target is to make protein. Okay, protein is very important in the cell's life, no, the cell survival. Okay, so now let's go into the DNA structure. There. So we'll start with the DNA. Now, the DNA is actually composed of 4 repeating nucleotides. Okay, I'm sorry for that. I'm sorry for the technical problem. Okay, I hope everybody will come back watching. Where is everybody now? Please message everyone that we are back online. Just wait a minute. Let me just message everyone that we are back. Okay, so our discussion now is on the DNA structure. As I mentioned earlier, the DNA is composed of 4 repeating subunits, 4 repeating nucleotides. These are the adenine, the guanine, thymine, and cytosine. So the adenine actually is a purine with a double ring structure, and in double-stranded DNA, it pairs with thymine. So adenine and thymine. Guanine is also a double ring structure. And in double-stranded DNA, guanine pairs with cytosine. So guanine and cytosine. And thymine is a single ring structure. So here, single ring structure. And the thymine pairs with adenine, and cytosine pairs with guanine. So these are our 4 repeating subunits of the DNA. Now, if we look into the DNA, the close-up of DNA, we got here a DNA that is unwound, so that we can have a look at it closely. So here you can see that our DNA has a backbone that is composed of an alternating sugar and phosphate. So where are those? Deoxyribose sugar and the phosphate group, and that's alternating. And then we can see that our DNA runs in 2 opposite directions. The 5' to 3' and the 3' to 5'. We can see also that between our nucleotides, either purine or pyrimidine, there are hydrogen bonds connecting them. And there are 2 bonds connecting thymine and adenine, and 3 bonds connecting guanine and cytosine. So, you just have to remember that. Okay. Let me just repeat it. Our DNA is composed of an alternating phosphate group and sugar group here. And these are our nucleotide, that the thymine and adenine with 2 hydrogen bonds, and our guanine and cytosine with 3 hydrogen bonds in the middle. Okay. So, that is our DNA structure. When we compare DNA with RNA, so we have like this. The RNA is just basically single-stranded, our ribonucleic acid, and our DNA is double-stranded, composed of cytosine, guanine, adenine, and thymine. Similarly, the RNA is also composed of cytosine, guanine, adenine, but instead of thymine, there is the uracil. So that's the difference between the RNA and the DNA. Okay, so we proceed. Now, very important to a living organism is its DNA to be replicated. If you remember last time in our discussion in cell cycle, we have mentioned that the DNA becomes single-stranded during mitosis or meiosis. And that is being duplicated in the interface part, the S of the interface. You have G1, the S, and the G2. And in the S phase, our DNA is replicated. So how does replication occur? Now, this I will tell you first the very basic principle in DNA replication. So here is a DNA replication fork. What happens in this replication fork? Okay. So DNA replication here begins when an unwinding enzyme breaks the hydrogen bonds holding 2 DNA strands together. I mentioned earlier that there are hydrogen bonds connecting the base pairs. So 2 between adenine and thymine, and 3 between guanine and cytosine. So there is, let's not talk about the names, what's the enzyme, what's the but you just have to remember that there is an unwinding enzyme that would break the bond between the bases. And that binding protein stabilize the DNA. Okay, so when it is unwound now, the enzymes called DNA polymerases recognize the primers as start tags. They join together nucleotide monomers between the primers. So exposed bases of a parent template dictate what will be the base, let me just repeat that part. What would be the pair that will be added into the sequence? So you just have to remember the base pairing rule, A with T and G with C and vice versa. So here is the exact process of DNA replication. And you remember earlier I mentioned that the DNA has two directions, 3 to 5 and 5 to 3. And our replication moves also in opposite direction. So in other strand, we have a continuous assembly, and in the other strand, we have a discontinuous strand assembly. In other books or in other discussion, they call this continuous strand the leading strand. Leading. So, napaka-simple para maintindihan, no? Because it's lead, so it means continuous. The discontinuous strand is the lagging. Lag. Diba tayo pag nagko-computer, pag may problema, bumabagal, we say, I lag. So, that's discontinuous strand assembly. So, the lagging strand. And we have to just still remember in here the base pairing rule, because in any direction, whether it's continuous or discontinuous, it will always follow the same pairing rule. And at the end, the DNA polymerases will fill in most of the gaps that the primers left behind. So, this is in the discontinuous strand. And then later on, another enzyme will come called the DNA ligases that will eventually seal the gap. So, the DNA polymerase fill in the gap. For example, there is a missing A, there is a missing C, and things like that. So, the DNA polymerase will put that in. But, to seal the gap, eventually so that it will become continuous this time, the DNA ligase will seal the gap. DNA replication is now complete. So, parang it's a bit difficult to picture out. So, I'll give you a summary of this DNA replication in this picture. So, here, we start here by having a double-strand DNA right here. Okay. And then you see this, the enzyme earlier that cleaves or unwinds the DNA, it's called the helicase. This is an enzyme that unwinds our DNA. So we have now two open templates, the leading strand and the lagging strand. So here is the leading or the continuous strand. Continue yan. Hindi siya nagli-leave ng gap. Walang naiiwan. Hindi siya pira-piraso or by strand. Itong strand muna na to, itong strand. Hindi ganon. Basta leading strand, tuloy-tuloy ang kanyang pag-read ng sequences and adding of the complementary pair. But in the lagging strand, there is the formation of Okazaki fragment. That's sounding Japanese because it is Japanese. No? Okazaki fragment. That Okazaki fragment is the fragment by which the discontinuous strand is adding into the template. So, di ba dito ang template, itong ating yellow here, and the replicate is itong green natin here, pero hindi siya continuous, putol-putol siyang inilalagay. So, by fragment. Ang tawag natin doon ay Okazaki fragment. So, pag nabuo na yung base complementary rule, yung DNA polymerase, lalagyan niya yan ng kompletong rule, pairs, kompletong pairs. Tapos, the DNA ligase will seal the gap. So, wala nang gap habang nagmo-move itong DNA polymerase at ang DNA ligase papunta sa direction niyan. So, the direction of this can be opposite. No? It can be 3' to 5', and the other one can go 5' to 3'. Okay. So, that's just very simple. All you have to remember in DNA replication is the base pairing rule, that when there is A, you have to pair it with T. And when there is T, you pair it with A. When there is C, you pair it with G. And when there is G, you pair it with C. So that, by example, we can have something like this. Okay? You have here the template. Here, the letters in black color. So, you have here a T, and our replicate following the base pairing rule is A. and then T with A, T with A, G with C, A with T, A with T, T with A, C with G, G with C, A with T, and until the whole strand is complete. So that is DNA replication. So I hope you get the idea. So what we are discussing here basically are just the principles. The other details, you just have to forget about that now. When you understand that the DNA is replicated in this manner, it will be easier to relate kapag ka meron na dyan yung details. Anong enzyme yan? Ano ang nagki-cleave na enzyme? Ano ang nangyayari afterwards? So, just remember the base pairing rule. Now, next part of our discussion here as part of the central dogma of life is transcription. So, transcription is the first of several steps of DNA-based gene expression in which a particular segment of DNA is copied into RNA, called the messenger RNA. So, if you remember earlier when I said that the central dogma comes from the DNA that is transcribed into messenger RNA and then eventually translated into protein. So, a part of that is the transcription having a product of messenger RNA. So, when you are asked, what is the product of transcription? Your answer would be messenger RNA. Or, I forgot to mention earlier, but there are actually 3 types of RNAs participating in here. No, 3 types of RNAs. The first one is our messenger RNA. The second one is the transfer RNA. And the third one is the ribosomal RNA. Now, you will see later what are the functions of these 3, especially our messenger RNA. Now, transcription is an essential step in using the information from genes in DNA to make proteins. Well, proteins are the key molecules that give cell structure and keep them running. So, now I will show you how transcription happens. So, transcription happens when a double-stranded DNA has the promoter region, and this promoter region will be recognized by the enzyme. So the enzyme starts attaching to the promoter region and it goes to the whole strand of the DNA so that the DNA will unwind. There, pag nag-unwind na siya, open template na siya, pwede na magkaroon ng transcription. So, in transcription, you have, okay, let's pause that. So, we have the sequence here, for example. Remember earlier in what I mentioned in the yung components ng RNA, we said that there is no more thymine but there is uracil. So itong blue, the blue strand in here is our template strand of DNA. So you have that sequence, and the growing RNA transcript, that's the messenger RNA, comes in this manner. So T with A with U, not with T but with U. C with G, C with G, and A with U. So that's what you are to remember. Pareho lang sa DNA, ang base pairing rule ay C at saka G. Yung A, ang kapares niya ay U. Wala ng T. Okay? So, that's the elongation part where the growing RNA transcript has this strands, or how has this base pairing rule. Okay, so A with U, T with A, and A with U again. So that's what happens. And eventually they will separate, and now we have a complete message that is ready to be deciphered for our protein synthesis, or in the process called translation. So take note of this. Paano ba itong binabasa? Paano ba natin maiintindihan? Paano ba magiging protein synthesis? Paano ba si-synthesize ang protein with only the strand that is what we have here, like this messenger RNA. Paano ba itong ginagawa? So we have to know how to decipher. And that is deciphered through the genetic code. This is our table of the genetic code that has the list of all the amino acids. So you know that amino acids are the building blocks of protein. So when you put together in long polypeptide chain, or many amino acids together, you are basically forming a protein. So how does that happen? May mga codes, no? Combination of 3. Pwedeng UUA, CAG, UCA, and like that, right? Diba? Meron kayo niyan in your modules. So here, I will show you combinations. For example, ano ba ang mga combinations that would give us DNA? So here is an interactive chart of deciphering messenger RNA showing the genetic code. So you have the uracil, the cytosine, adenine, and guanine. But there are only 3 for each code. So say we have U, C, and A. What does that give us? Serine. No, it gives us the serine. What about C, A, G? That gives us glutamine. Okay? Another example, A, G, and U. Another serine. Okay. So you can have all the combinations in here, and malalaman natin like ACU, threonine, ano pa ba ibang example, GAU, that's aspartate. And we also have the stop codon, or tawag natin dito codon, no? Resulting codon. Take note that there are a total of 64 codes in the genetic table. 61 are actually coding for amino acids, and 3 are stop codon. So you have here, stop codon. So ano ba ang tatlong stop codon? You have the UAG, UAA, and UGA. So ako, para mabilis ko yan matandaan, sinasabi ko uga, uwag, and ano na nga yung isa? Ano yun? Stop dito. U-A-U. O-A-A. O-A-A. Yan. Gumagawa lang ng mnemonic so that you can easily remember. Don't worry, I'm not gonna make you memorize the genetic table. That's why it's always provided. Okay? I hope you understand transcription. So in transcription, all you have to remember is that your DNA strand is now an RNA. Now, when it is read, it becomes an RNA. And that RNA is what we need in the next topic, which is translation. Ayan. So translation is the second process of protein biosynthesis. And this is part of overall process of gene expression. In translation, this messenger RNA is decoded to produce a specific polypeptide according to the rules specified by the genetic code. Translation is necessarily preceded by transcription. So, protein synthesis will not be complete kung transcription lang, hanggang messenger transcript lang tayo or RNA transcript. That transcript must be translated. So, a cell reads information from a molecule or from the messenger RNA and uses this information to build a protein. Translation is happening constantly in normal cell. So, protein synthesis is very important. You remember in our topics last time, even in the cell wall of the cell or the cell membrane, you see that proteins are participating in membrane transport. No? They can act as carrier protein, the gated protein, things like that. So proteins are very important. So how does translation happen? Now let me give you a very quick overview of the translation. So first, in translation, the other two types of RNAs are participating. And these are the transfer RNA. So this one here is a transfer RNA that carries the amino acid. So siya ang taga-transfer ng amino acid following the codon in messenger RNA. So ang dala-dala ng transfer RNA ay anticodon. Yung nasa messenger RNA ay codon. So, dala-dala niya yung specific amino acid, yung galing dito sa codon table natin. Leucine, isoleucine, valine, threonine, and so on and so forth. So, ang nagdadala niyan is our transfer RNA. So, it has the anticodon to the codon of the messenger RNA. And also, the small ribosomal subunit and large ribosomal subunits, or these are the rRNA, ribosomal, RNA, are also participating. So, how? Tingnan natin. This is my simplest discussion of the protein synthesis. You know, when I was a student, it was really a struggle for me to understand central dogma. You know, from replication to transcription to translation. Kasi nung araw, sa amin, we don't have mga animated videos to show kung paano nangyayari ang mga bagay-bagay. So, nowadays, we have a lot of resources, and to make it simple, let's see this translation process. So, translation proceeds through 3 stages. You have the initiation, the elongation, and the termination. Now, this happens when the messenger RNA, the small ribosomal subunit, and the large ribosomal subunit of the ribosomes come together. Okay? So, sila yan, yung large ribosomal subunit, small ribosomal subunit, the messenger RNA, and the transfer RNA. Itong 4 na to, pag nagsama-sama sila, ang tawag natin dito ay initiation complex. So, here in the figure, you can see in our initiation complex, meron tayong binding site. The binding site for the messenger RNA and the binding site for the amino acids which are carried by the transfer RNA. So, example here, meron tayong messenger RNA na nakalagay dito sa taas, no? And then, babasahin siya ng transfer RNA carrying the anticodon for the amino acid. So, meron tayong A site, P site. Ano mangyayari after that? You have the start codon. Usually, ang start codon is the code for methionine. Ayan. And then, another one to the anticodon. Ayan, matatanggal, magdidetach ang amino acid. Magpo-form ng chain ang amino acid to another amino acid. Itong transfer RNA ay matatanggal. lilipat siya sa kabilang site, and then the elongation will continue. So, yan na yung elongation ngayon, no? Until matapos basahin ng transfer RNA ang message in the messenger RNA. So, you have now a growing polypeptide in the platform. So, pag meron ka niyan, mag-stop yan siya, class, kapag ka nakaload ang stop codon. Okay, so ano nga yung mga stop codon natin? Kung ang dulo ng messenger RNA ay UAA, UAG, UGA, yan, mag-stop ang protein synthesis or ang translation. So, let me just repeat this animation. Okay? So, showing in here the small ribosomal subunit. And then you have the initiator, transfer RNA, coming together. Next to come is our messenger RNA. When the messenger RNA is already there, the large ribosomal subunit will come, and they all form our initiation complex. Yeah. So once we have initiation complex, we have now the translation now can proceed. So showing here the sites, binding site for messenger RNA, and the P site and the A site for our transfer RNA. So reading the messenger RNA, the initiator transfer RNA will bring in the amino acid that is coded in the codon. So AUG is actually for methionine, but it is also a start codon. And then the CAG will be the anticodon for GUG, and that brings another amino acid. And this process will continue. One transfer RNA will be dislodged, and another transfer RNA will replace it, following, of course, the rule, the base pairing rule. And then, so amino acid will form the polypeptide chain, so it will grow longer, and the chain will stop until the stop codon is loaded into the platform. So when there is the stop codon, the messenger RNA will be detached and the translation is now complete. So this is the process of translation. Okay. Now to sum it up, I just would like you to see what happens in the whole of the protein synthesis. So here, protein synthesis is a two-part process that begins with transcription. Now after, that's the DNA replication. So transcription in the nucleus. In the nucleus, genes that encode messenger RNA, ribosomal RNA, and transfer RNA are transcribed. Naka-transcribe silang lahat into messenger, into ribosomal, and transfer RNA. Then, ribosomal RNA combines them. The RNAs and the ribosomal subunits exit the nucleus through nuclear pores and enter the cytoplasm. So that here, the three classes of RNA interact in translation, which is the synthesis of polypeptide chains for free amino acids. So you have here the long polypeptide chain of amino acid forming protein. Ayan. Okay. So now we are ready to have a short exercise. Are you ready? Okay. So I have a short quiz here for you, and this one with a prize. Whoever put in our chat the correct answer, the first one to give the correct answer, nag-stammer na, to give the correct answer, will get a prize. Okay, so we have a DNA sequence here, CAC, GTT, CGA, ATC, CGA, ATT, ATG. What is the DNA replicate? What is the DNA replicate? I'm waiting for your answers. Sino kaya ang unang magchat-chat ng tamang sagot? Oh, ang bilis naman ni Christine at ni Maricar. O, si Christine ang pinakauna. Tingnan natin. Ang sagot ni Christine, si Maricar at si Jillian ay hindi magkapareho. Bakit? Okay, ang tamang sagot ay natin you have the CAC. So, ang tamang sagot, tingnan nyo kung tama ang sagot nyo, ha. GTG. Tama? GTG. CAA. GCA. GCT. Sorry, GCT. TAG. Tama ba ako? Saan na? GCT. Tapos TAA. And then TAC. TAC. Oh, tama si Christine. I'm gonna message you later, Tin, for your prize. Okay, sino pa may mga tamang sagot? Let me see. Tingnan ko muna mga sagot nyo, ha. GTG, CAA, GCT, TAG. Ang gagaling nyo, ha. Yung iba siguro mga typographical error na lang yung na-type na iba sa sobrang pagmamadali. Wow, many of you got the correct answer. Ibig sabihin, nakinig. I'm so happy. You have ang mga sumagot, si Christine, si Maricar, si Jillian, si Lay, Angelica, John Michael, Almira, Jofet, Anna Joy, Jeanette, Jamaica, Romy. Bakit parang kulang ang strand mo, Romy? 1, 2, 3, 4, 5, 6. Kulang ng isa. Isang triplet. Maria Leia. At I will print screen muna nito para makita ko yung inyong mga sagot. Ayan. Okay. Wow, ang gagaling. Okay. So now, take note of your DNA replicate, ha? Ang DNA replicate ninyo because that is where we are going to base that is where we are going to base our next question. If you have that DNA strand and then that DNA replicate, what is your messenger RNA based on the replicate? Based on the replicate. I'm waiting for the answer. This time, this is messenger RNA, okay? Messenger RNA. So, what is your messenger RNA based on the DNA replicate? Ang bilis naman talaga ni Tintin. Nangunguna parate. Yung iba kasasagot pa lang ng DNA strand. Oh, meron ng sagot. Tingnan natin ang sagot mo, Tin, kung tama ba. Wala? Ba't nawala? Retracted the message. It's okay, Romy. Nakulangan. O, ayan. O, sige. Nasaan na? Oh, si Maricar. Let's say, Maricar, kung tama ba ang iyong sagot, our answer must be CAC. Oh, correct. CAC. Ang next is GUU. Oh, correct. Next is CGA. CGA, tama. And then, AUG. Ala, bakit sa iyo AUC? Sayang. Kinulang. AU. Ha? Ako ba ang mali? Ay, mali ako. 1, 2, 3, 4, 5. Oo nga. Teka lang. I have to edit. Ako palang mali. C, G, A, T, A, T, G. So, dapat T, U. Sana yun, Maricar, na wala na. Ha? AUC. Nanaman ako. TACG. So, ang replicate mo ay may tag. Tapos ito, ang sunod dapat nito ay AUC. Bakit ganun? AUC. Hindi, parang hindi to Maricar yung ano. Tama. Bakit naging UAC na yan? AUC? Dapat? Oh. So, sino na unang tama? Tingnan daw natin ang kay Jofet. Baka si Jofet ang tama. CAC, GUU, CGA, tapos AU, hindi AUG. Wala, tinatanggal na nila yung kanila, retracted the message. So, tingnan natin tong, kay Jillian. Baka tama si Jillian. Anong CUC? Bakit nag-CUC ito? Ha? Walang C. Ang T, A pa rin ang kapartner niya. Yung A lang ang kapartner niya, U. So, dapat, C, bakit kayo ganyan? Bakit ganyan ang mga sagot nyo? Teka, makinig kayo. Ito ang tamang sagot, ha? Ito ang tamang sagot. Sige, mag-type nga daw kayo ng tamang sagot. Bakit ka sumasama? Ang tamang sagot ay ito. CAC, parang may mali ako dito eh. Ito kasi dapat, ito ay G. So, dapat ito ay C. Okay, yan ang tamang sagot. CAC, GUU, CGA, AUC, CGA, AUU, and AUG. Sino nakatama? AUG, CAC. Bakit maging CUC ang Jillian ang sa iyo? Bakit naman sa iyo, Jeanette, naging GAC? G na nga yun, oh. CAC. Baka si Romy ang tama. Ay, madami na sa baba. Madami na sagot. Teka lang, binalikan ko. So, Romy, ang sagot mo ay CAC, GUU, CGA, AUG. Oh, kasi may mali ako kanina eh. Ako yung may mali kanina. So, baka yun ang pinagbasihan ni Romy eh. AUG, tapos CGA, AUU, AUG. Oh, I recognize. Romy, tama ikaw. Sige. Kasi alam ko kanina kaya naging AUG ang sagot mo ay dahil ako yung nagsulat ng TAC dito sa replicate. Ang saya naman. Ang gagaling nyo, class. I'm so happy. Halos tama ang inyong mga sagot. Also, may mga typographical error lang. Malamang dahil lang sa pagmamadaling sumagot. Aray. Okay. Ano bang nangyari dito? Ang sunod na question is... ano ba yan? Nakikita nyo pa ba? What is the transfer RNA? Ano ang transfer RNA? Let me see. What is your transfer RNA? Transfer RNA, class, is the other term for that is anticodon. Sino makakasagot ng transfer RNA? Oo, yes, Joy, tama ka. Kasi takpo kasi nakasulat kanina kaya ang sagot nyo AUG. I understand. Yes, that's okay. That's considered. Sige, I'm actually taking photo of your answer para mabalikan ko mamaya and I can give credit points to those who answered correctly. Pero meron na akong dalawang unang ang pinakaunang tama kanina is si Christine, tapos ang pangalawa ngayon ay si Romy. Sa transfer RNA natin, transfer RNA. May sumasagot na ba ng transfer RNA? Transfer RNA, nabayan yung sa iyo. Si AC, GUU, hindi pa kay Giselle. Ang sa iyo, Lay. Transfer RNA. Hindi yan, kasi may T. Sino na nga, Lensky ba ito? Ikaw ba to, Jorilyn? GUG, CAA, GCU, teka lang nga wala, GCU, UA, G, and then UAA, okay, mukhang tama si Jorilyn. So, tingnan natin ang sagot. What is the transfer RNA? Transfer RNA natin ay, balikan ko lang yung nagkamali din ako kanina, no? So, dahil ito na GCTA, TAAU, CG, GC, CG, TA, TA, UU, AUG. Tama ba ko? Okay. So, ang tamang sagot natin ay GUG-CAA. Nga ba yung sagot mo? Jurilyn? Saan na yun kanina? Pinakauna. Yes po, ma'am. Si Jurilyn. Oh, wait lang. Hanapin ko muna uli. Saan na ba ito? GUG-CAA, GCU-UA, G, C, UAG, and then GCU, UAA, UAC. Tama. So, correct si Jurilyn. Siya ang pinakaunang nag-type ng tamang sagot for our yung kay CAC. Hindi. Okay. So, meron na tayong, tama no, si Christine, si Romy, si Jurilyn. Next question. What is the amino acid? Tingnan ko nga kung nakinig ba kayo kung ano ang magiging amino acid. Amino acid. Hala. Meron ba kayong hawak na ano? Teka lang. Wala pala akong nilagay na ano ay. Yung genetic table. Kung may mga hawak kayong module, have a look, parang mas madaling tingnan sa inyong mga module kaysa ganitong table. Okay? Anyone with an answer? Oops. Print screen so that I will know who answered. Wala pang sagot? Amino acid. Valine. Oh, si Maricar. Tingnan natin. Valine, glutamine, alanine. Stop. May stop tayo? May stop. May stop ba kayo tayo dyan? Oh. There is something that I must discuss again. Valine, glutamine, alanine, stop, stop, stop. Oh. Okay, stop muna. Stop muna sa pagsagot ng ng amino acid. I think you are getting it wrong. Maybe I was not able to emphasize it earlier, but stop for now. Okay? Now, I would like to tell you one thing. Stop first, okay? So, here, our DNA template. The DNA replicate is based on the DNA template. I mentioned that the messenger RNA is based on the DNA replicate. And transfer RNA is the anticodon. Pag sinabing anticodon, that is based on the codon. So the codon is the messenger RNA. But class, amino acid is based on codon, not anticodon. So, you will have to read the messenger RNA, not the transfer RNA, in order for you to get the correct sequence of amino acid. Amino acids are based on the messenger RNA, not on the transfer RNA. So, you do it again. What are the amino acids of our messenger RNA, of our codon? No, codon yan siya, ang amino acid. Okay, I'm waiting. Wala pa, may mga stop-stop pa kayong sinulat eh. So, that means that's not yet it. Based on okay. Let me just wait for your answer. CAC, histidine, kalito po ma'am, sorry. That's okay, Mark. So, sino may sagot? Oh, wait lang ako. Sige, don't worry. Let me see. Kung tama ba ang sagot nila? Ito. Kay, sino bang nauna? Si Maricar. Histidine. Next is valine, arginine, and then isoleucine, arginine, phenylalanine, methionine. Arginine, isoleucine. Okay? So, here is the answer. Of course, meron tong na-change dito. Sorry. Let me just edit this. And then this would be C. Okay. And this would be G. So, AUC, I think, is for AUC. A, U AUC isoleucine. Isoleucine. Okay, are you correct? And then arginine. Tama si. May phenylalanine. Bago ang methionine. AUU. What's AUU? AUU. Isoleucine man ang AUU. AUU is isoleucine. Bakit may phenylalanine kayo? O, sino may tamang sagot? Walang stop. Ay, oo nga. Itong AUU, walang stop. AUG. Walang stop, Arnel. Bakit may phenylalanine kayo? Tama ba yan? Si Jillian at saka si Maricar ay pareho ang sagot. Let me just check that later. Oo nga, may phenylalanine talaga. Phenylalanine? Saan ang phenylalanine? Yung AUU? AUU is phenylalanine. AUU. Bakit? Isoleucine. AUU. Wait lang. Dito ako titingin sa aking module. Okay. AUU? Hindi man phenylalanine ng AUU? Isoleucine. Did anyone get it right? Oh, sino na may tamang sagot? Si Almira. Histidine, valine, arginine, isoleucine, arginine, isoleucine, methionine. I guess that's the correct answer. Walang phenylalanine. Sino na una? Si Jamaica. Arginine. Si Kim. Marami na palang nauna. Wait. Si John Michael. Tama ang sagot niya. Pinakauna kung sino na unang maglagay ng tamang sagot. Hindi ilusin ang kay Kim. Teka. Histidine, valine, arginine, isoleucine, methionine. Angelica, that's not methionine ha, methionine ba? Ang merong proline, walang proline, John Michael. histidine, valine, arginine, isoleucine, arginine, may phenylalanine. Bakit iba-iba masagot ninyo? Tama si si Angelica May. Although, hindi lang not-spell ng maayos ang methionine. Okay, anyone? Do you have any question? Anything that you would like me to discuss again? Anything that you did not understand properly? Anyone? I'm waiting for your... Any question, class? Madami ang tama. Mayroon na nalilito lang doon sa ibang Okay. Any question?